Hey friends, welcome back to my channel Rajni Bio Academy. So in last lecture we discussed hydropathy plot. So let's continue with its application. So hydropathy plot helps in prediction of transmembrane helices and gives protein topology, whether proteins are 2D, 3D, alpha helices or beta sheet, basically structure. So hydropathy plot used in bioinformatics tool, example kite Doolittle method. So next we have protein fracture technique. It is a microscopic preparation technique used to study the structure of biological membranes, especially lipid bilayer and membrane proteins. So it is based on the principle that the fact that membranes split along the hydrophobic interior of the lipid bilayer, so the two leaflets separate, reveals internal organization of membranes rather than surface view. So if we have to study it from inside, then we use this technique. So steps involved: first one, rapid freezing. So it is similar to cryopreservation. Sample is frozen at -196 degrees Celsius at liquid nitrogen. So this prevent movement of molecules or kind of immobilize them. Next step is fracturing. Sample is cut using cold microtome knife. So fracture occurs along lipid bilayer, separating it into P phase, protoplasmic phase or inner leaflet, and the second one E phase, exoplasmic phase or the outer leaflet. Next step, etching. So slight warming, we can say ice sublimation, or we can use vacuum drying. So exposes more the structural details. Now next step is replica formation. So surface is coated with heavy metals like platinum or carbon. So biological material is removed and it leaves a replica. Observation: replica is viewed under electron microscope. So key observations are membrane proteins appear as intramembranous particles, IMPs, and there is unequal distribution of proteins on P surface and E surface. So there is unequal distribution on the outer leaflet and inner leaflet. Application: study of membrane protein distribution, cell junctions, organelles membranes like mitochondria, ER, etc. So our next topic is transport proteins. Classification of membrane transport proteins based on the function. There are two types: carrier proteins and channel proteins. So all transport proteins are multipass transmembrane protein. This is important. Okay, so let's start with carrier proteins. So they are responsible for selective transport via conformational change. So carrier proteins are of two types. First one, uniport. They transport single solute. They are unidirectional, no coupling with other solutes. Example, glucose transporter. Second type, co-transport or coupled transport. So transport of one solute is linked to another. So it is further divided into two, symport and antiport. Symport, both solutes move in the same direction. Example, glucose, sodium glucose transport. So they are moving in the same direction. Antiport, solutes move in the opposite direction. Example, sodium calcium exchange. So they are in the opposite direction. Next one is channel proteins. So channel proteins, they form pores for solute movement. Transport occur down the concentration gradient and always passive transport is there, faster than carrier mediated transport. Next is types of channel. So first one is leak channel or non-gated channel, always open and allow continuous ion movement. Second type, gated channel, open or close in response to signal. So gated channel is further divided into voltage gated, open due to membrane potential charge, like this. Second type, ligand gated, open by ligand binding. Ligand may be extracellular neurotransmitter or intracellular ion or nucleotide. Third type, mechanical gated, open due to mechanical stress. Fourth one, ion gated, regulated by specific ions. Fifth type, nucleotide gated, controlled by cyclic nucleotide, cyclic AMP and cyclic GMP. So important point to remember, channel proteins form pore for passive transport. Some are always open like leak channel, while others open or close in response to specific signal. So here is the difference between carrier and channel transport protein. So mechanism, here conformational change take place. Over here in channel protein, pore formation is there. Speed, carrier transport protein, they are slow, channels are very fast. Transport, active or passive both. In channel protein only passive. Saturation, it is there, and in channel no saturation. So types of carrier protein based on transport mechanism: uniport, this is single solute, and co-transport, symport or antiport. So over here is a chart representation of transport proteins. Important points to remember: carrier proteins bind the solute and undergo conformational change. Channel proteins form pores for passive transport. Carrier protein can be saturated, channel proteins generally cannot. Channel may be open, leaky or gated by ligand, mechanical stress or voltage. So that's all for today. See you in the next lecture.